Welcome to Microbes Unveiled, a brief exploration of the hidden world of microbiology. The cover image and side panels show diverse microbial forms and set the tone for what follows, introducing themes of structure, function, and application as we move from basic cell types to engineered microbes and therapeutics. This slide compares prokaryotic and eukaryotic cellular organization using side-by-side diagrams, highlighting that prokaryotes lack a nucleus and compartmentalized organelles while eukaryotes contain nuclei and specialized internal membranes. These structural differences shape function, evolution, and how antibiotics target unique features of microbial cells. Here we see how bacteria replicate by binary fission and how growth proceeds through lag, log, stationary, and death phases. The charts illustrate exponential doubling under optimal conditions and show that nutrient availability, waste accumulation, and environmental stress determine population dynamics and practical outcomes for culturing or infection control. This slide summarizes sterilization strategies, contrasting physical methods like autoclaving, filtration, and UV irradiation with chemical agents such as alcohols, halogens, phenolics, and aldehydes. The visuals emphasize how different approaches inactivate proteins, membranes, or nucleic acids and why defined kill times and method selection matter for safety and efficacy. The virulence factors diagram walks through how pathogens attach, invade, and evade the host, from adhesins that colonize tissue to invasins that penetrate deeper, capsules that block phagocytosis, endotoxins that trigger systemic inflammation, and exotoxins that disrupt cells. Collectively, these mechanisms determine infection severity and clinical outcome. Antibiotic resistance arises through multiple neutralization pathways shown here: enzymatic degradation, target modification, efflux pumps, and metabolic bypass, all of which can be shared between microbes through horizontal gene transfer via plasmids and transposons, accelerating spread and resulting in ineffective therapies that undermine standard antibiotic treatments. The colon microbiota functions slide maps dietary inputs like fiber and nutrients to microbial activities: vitamin synthesis, fiber fermentation producing short-chain fatty acids, pathogen exclusion by competitive colonization, and training immune tolerance. These processes link directly to metabolic and mental health, showing how microbial balance supports overall well-being. Selected probiotic strains such as Lactobacillus and Bifidobacterium are highlighted with measurable clinical endpoints, demonstrating benefits like reducing antibiotic-associated diarrhea, modulating cytokine profiles toward balanced inflammation, strengthening the intestinal barrier, and even decreasing infant colic, all supported by randomized trial data for specific outcomes. Microbial cell factories in action shows practical applications. Yeasts convert sugars to ethanol and carbon dioxide for brewing, Lactobacillus produces lactic acid for dairy preservation, and engineered E. coli can secrete therapeutic insulin. The common theme is scalable production under controlled bioreactor conditions to deliver consistent, useful outputs. Engineered microbes for renewable energy are illustrated converting sunlight and CO2 or plant biomass into biofuels. Cyanobacteria engineered to make ethanol or butanol, and cellulolytic Clostridia breaking down biomass for biodiesel precursors, with the dual benefits of mitigating greenhouse emissions and reducing dependence on petroleum feedstocks. CRISPR engineering enables microbial logic for biosensors, living therapeutics, and programmable kill switches. The diagrams show sensor systems detecting pathogen nucleic acids, engineered microbes delivering anti-inflammatory molecules in vivo, and built-in safety switches that trigger lysis under defined conditions to ensure biocontainment and controlled therapeutic deployment. Next-generation microbiome therapies combine multiomics data and AI-driven strain selection to create personalized microbiome transplants, targeting conditions like obesity, depression, and autoimmune disorders. The visual flow emphasizes precision health outcomes by restoring ecological balance and tailoring interventions for measurable, individualized wellness benefits.